Excellent. Hello, everybody. Lovely to see you all joining us today for our second MINAGRIS webinar. Please do make yourselves comfortable, introduce yourselves in the chat. We'd love to hear from you. And we will get going in around 30 seconds. Good to see lots of people joining us today. That's wonderful. I hope that we've got people from all over the world, so please do say where you're from. We'd love to hear from you. If you have any technical difficulties or anything as well, please do also post in the chat to let us know. Hopefully you can hear me loud and clear, but do say if not. Okay, so we'll get underway. I can see we've still got a lot of people joining us, but we're keen to stick to time today. So today's webinar is going to be presented by Stathis Lagos and Eleni Lamprou. They are from the University of Thessaly in Greece. This is our second webinar hosted by MINAGRIS. So MINAGRIS is a 5-year EU-funded research project. We are looking at the impacts of micro and nanoplastics on agricultural soils. So during today's webinar, we're looking at what happens when microplastics meet agrochemicals. So these are two key emerging and past stressors that are building up over time. And what we're going to look at today is what happens when these stressors meet soil microbes and also how they affect nitrogen cycling. As I'm sure you're all aware, both of these things are very crucial for soil health. So I shall hand over to Eleni and Stathis shortly. One thing I'd like to make you all aware of first is that we are keen to invite you to our final conference. So we are hosting a final conference in person in Brussels on the 16th of September. It's free to attend, and I believe the registration link will be posted in the chat very shortly. It is also a hybrid event, so those willing to join online can also do so. We'd really like to encourage in-person interaction, though, as that's where all the buzz and excitement will take place. So without further ado, I'd like to introduce Stathis and Eleni. They will speak for around 40 minutes, give or take, and then we will have a question and answer session. So please do post in the chat throughout. Over to you guys.
Okay. Thank you very much for the introduction. Hello, everyone. My name is Eleni Lamprou. I am a PhD candidate at the Department of Biochemistry and Biotechnology here in Larissa. And today, me and my colleague Stathis will present you a part of my PhD work, which is in the frame of European-funded project MINAGRIS. And today we will discuss about the microplastics when made agrochemicals, their interactive effect on soil microbes and nitrogen cycle. So as you all know, in recent decades, plastic have become integral to modern life, with their annual production continuously increasing, marking the current era as the plastic age. As we see here in the graph, the projection for the usage of plastics from 2020 to 2060 in Europe raises from 400 to 1,000 million tons, while for America more than 1.2 million tons for the year of 2060. However, this excessive use of plastics leads, of course, to the production of huge amount of plastic waste. Despite our progress on recycling and plastic waste management in general, still millions of tons are released in the environment every year. Plastic pollution is found globally, from the deserts to mountaintops or to the deep ocean. This consistent plastic pollution represents a significant global threat to ecosystems, wildlife, and human health, fitting the profile of a poorly reversible pollutant. Based on their characteristics, plastics can undergo fragmentation due to weathering, photooxidation, biodegradation, and chemical degradation, giving rise to smaller pieces, namely microplastics. Microplastics are known for their small size, less than 5 millimeters, and considered as the smallest the medium size of the plastics. Plastics are released into agricultural soils intentionally or unintentionally. Deliberate sources include plastic materials in irrigation systems, greenhouse coverings, and mulching, while unintentional inputs came from plastic loads presenting compost, manures, coating seeds, fertilizers, or pesticides. Among the different plastic types often used in agricultural soils, low-density polyethylene constitutes the most common plastics. Considering the resistance of the LDPE and its accumulation in agricultural soils, biodegradable plastics have gained ground. PBAT-based and starch-based are the most produced biodegradable plastics globally, covering around 13.5% and 18.7% of the production respectively. However, as Fao mentioned, the land we use to grow our food is subjected to much greater pollution from plastic contaminants. So, soil serves as a major reservoir for plastics, with agricultural soils being a primary area of concern. Once plastics end up in soil, they can occur with different organic pollutants such as pesticides, veterinary drugs, or antibiotics. Plastics introduce a distinct set of environmental challenges due to their high specific surface area that enables the absorption of environmental pollutants, effectively concentrating these contaminants on their surfaces. These interactions occur through processes such as adsorption and chemical affinity, potentially altering the fate, transport, and bioavailability of these pollutants. So by serving as carriers, plastic can facilitate the distribution of these contaminants, posing risks to soil health and the broader ecosystem, resulting in synergistic toxicity. On the other hand, monitoring of the ecological effect of soil pollutants on soil microbiota relies on the assessment of inhibitory effects on microbial groups, having a pivotal role in ecosystem processes. Nitrogen cycle is a key biochemical process and is controlled by multiple groups of functionally diverse microorganisms. Ammonia oxidizing microorganisms such as ammonia oxidizing archaea and bacteria that catalyze the oxidation of ammonia to nitrite have a rate-limited role in nitrogen cycle. While Comomox bacteria have been discovered in 2015, and it's a group of microorganisms that are able to complete the total conversion of ammonia to nitrate in one step. Nitrate, the end product of nitrification, is further transformed by a diverse group of heterotrophic facultative anaerobic bacteria into dinitrogen. This process is called denitrification, and if it's incomplete, leads to release of nitrogen oxide, which are toxic or highly potent greenhouse gases. Regarding the above, we turn our attention to investigate firstly the effects of different types of microplastics, both conventional and biodegradable, on the soil dissipation of albendazole, which is an anthelminthic veterinary drug, and pyraclostrobin, which is a widely used fungicide, in bipartite and tripartite co-occurrence scenarios. And secondly, determine the individual and interactive effects of microplastics with albendazole and pyraclostrobin on soil microbial functioning, using ammonia oxidizer and denitrifying bacteria as indicators of perturbation of nitrogen cycle in soil. To evaluate these points, we conducted microcosm experiment in 3 different soils. Today, I will show you the results of Chania soil. It's a sandy soil originated from Greece, in Crete, here in Greece. It's a Mediterranean region that is a hotspot of climate change. In this point, I would like also to mention that all soils selected among a range of other agricultural soils around Europe based on their negligible background contamination levels with microplastics. The soils treated with three different microplastic types, the LDP, PBAT, starch-based, but also the mixture of those. And they applied in two different concentration levels, 0.01% and 0.1%, either alone or in combination with albendazole and pyraclostrobin, and we further incubate the microcosms for 90 days to 20 degrees. At the specific time points, we determined the dissipation kinetics of albendazole and pyraclostrobin in the presence or absence of three types of plastics and calculated the DT50 values. Furthermore, the effects of the nitrification activity were determined through measurements of potential nitrification rate and nitrate concentration, while effects on the abundance of ammonia oxidizer and nitrified bacteria were determined via qPCR of relevant microbial gene markers like AMOA and NIRES. Moving to the results, here we can see the dissipation curves, and as you observe here, they can differentiate through the treatments. First, we can see the dissipation curves of albendazole and its transformation products. Secondly, albendazole in the presence of pyraclostrobin. And at the last graphs, we can see the dissipation curves of pyraclostrobin either alone or in the presence of albendazole. But we will focus better on the DT50 values on the next slide. So here in this slide, we can see the table with the DT50 values of albendazole and its transformation products when applied alone on the left side and in conjunction with pyraclostrobin. Here we noted that in the presence of any type of plastic, albendazole and its transformation products were accelerate the dissipation, while dissipation of total residues of albendazole was delayed in the presence of pyraclostrobin. Regarding the dissipation of pyraclostrobin, we didn't observe any significant alteration on the DT50 values affected from the microplastics. However, the dissipation of pyraclostrobin was inhibited by albendazole regardless of the presence of microplastics. Moving to sum up for our initial results, we demonstrate that microplastics can influence the dissipation of chemical compounds, but this effect is compound specific. In other words, in the presence of microplastic does not affect all substances in the same way. The extent and direction of the impact depend on various parameters, including environmental conditions and physicochemical properties of the compounds. Furthermore, we observed that interaction between co-occurring pollutants play an important role in shaping dissipation dynamics. When multiple contaminants are present in the same time, they can influence each other's behavior, altering degradation rates and pathways. Finally, our findings provide clear evidence of complex interaction between pollutants and microplastics, and this interaction highlights the need to consider multifactorial processes when assessing contaminant fate in environments where microplastics are present. Now I will give the floor to my colleague Stathis.
So, hello everyone, by me as well. I'm Stathis Lagos and I am a postdoctoral researcher, member as well as Eleni of the Plant and Environmental Laboratory in the Department of Biochemistry and Biotechnology of University of Thessaly. And now I will move to the results regarding the combined microplastics and pollutants influence on nitrogen cycle. As Eleni mentioned before, we studied three different soils. one from Chania, Greece, which I will call as Greek soils, Greek soil from now on. One from Dijon, France, so-called the French soil, and one from Wageningen, Netherlands, so-called the Dutch soil. In general, during the next slide, I will focus on the results regarding the Greek soil. There is no kind of chauvinism here, I want to assure you. We observed effects also in the other two soils, but in most cases they were scattered, time point or treatment specific, without showing any clear pattern or trend. We start with the effects on potential nitrification rate, which provides a functional indication of nitrification activity through the measurement of nitrate production. But before describe the results, two things I want to add here in order all of us to be on the same page. First, the 30 and 90 days you are going to see in most of the results I will show you, referring to the sampling time points where we assessed early and late exposure effects, respectively, in nitrogen cycle. Secondly, in all graphs like this one, I will present you with the combined presence of organic pollutants and microplastics. Only the data from the high dose rate of microplastic is presented. This is for enhanced clarity of the results, since our preliminary analysis showed no significant difference in the measured endpoints across the different dose rates of microplastics. So back on the results now, I have cleared everything. As you can see in the graphs here from the Greek soil, as I mentioned, when microplastics were combined with pollutants, we observed a clear, significant reduce on nitrate temporal levels in all treatment where microplastic coexisted with albendazole. And this effect was observed for both time points, but it was more evident during at 30 days. Moving now on the effects on nitrate levels, we observed a very similar pattern, again in the Greek soil. Co-application of albendazole and microplastic significantly led to significantly reduced nitrate levels. But in this case, this reduction was much more clear and evident also after 90 days. This is important because, as Eleni mentioned before, nitrate is the, let's say, end product of nitrification. So the shift we observed before with the functional screening of potential nitrification rate are reflected in measurable change in nitrate levels. However, if we zoom in just to the effects of microplastic alone without pollutants, a different picture emerges where starting with potential nitrification rates, we see that microplastics alone induce a significant temporal increase in potential nitrification rates at day 30, both in Greek soil, as you see, and in this case also in case of the Dutch soil. In addition, this pattern was followed and it was evident in case of nitrates, but only in the Greek soil, where the level also remained significantly increased compared to the control, of course, but in this case up to 90 days. So this suggests that microplastics alone may transiently have the ability to stimulate somehow nitrification process. Now we move on to the effects on the abundance of the ones who are actually responsible for nitrification process by targeting ammoate gene, as Eleni mentioned before, and starting with the ammonia oxidizing archaea, where again in the Greek soil you see here on the graph that albendazole appear again to be the main inhibitory driver at 30 days, reducing ammonia oxidizing archaea abundance across all microplastic treatments. However, at 90 days, we observe a recovery on their population in most of the treatments, suggesting maybe a temporal dynamic of these effects or an adaptation mechanism of this group. Looking now at ammonia oxidizing bacteria abundance, we observe the same but more persistent pattern. Again, we see here the results at 30 and 90 days from the Greek soils, where combined exposure to albendazole alone or with the presence of pyraclostrobin and microplastic resulted again in a sustained, at this time, reduction of ammonia oxidizing bacteria abundance. As for common bacteria now, in Greek soil, we observed a response pattern which is closer to ammonia oxidizing archaea than ammonia oxidizing bacteria, so an early inhibition followed by recovery of their population at 90 days in most of the microplastic treatments. So overall, we can see that the observed temporal or persistent decline in abundance of all ammonia oxidizers microorganisms in treatment where albendazole was co-present with microplastics, they are completely in line with the previous observation on potential nitrification and nitrate levels. However, again, when focusing only on microplastics presence without the pollutants, we observed some interesting contrasting responses between ammonia oxidizing archaea and ammonia oxidizing bacteria. Again, I'm referring to the Greek soil now. As you can see, in all microplastic treatments where we observe a significant decline of ammonia oxidizing archaea abundance, at the same time we observe a significant increase on ammonia oxidizing bacteria abundance. This pattern was present in both time points, but it was much more evident after 90 days in every treatment, as you can see on the graph. This suggests that maybe microplastics could influence niche dynamics among ammonia oxidizer, potentially playing a role on the restructure of this community. In contrast now, when we examine the nitrifying bacteria by targeting the nearest gene, the responses were in general much more stable. One of the few notable observations was the transient positive interactive effect in the Dutch soil at this time under the presence of pyraclostrobin and specific microplastic types, especially PBAT and starch-based, both considered biodegradable, but this was not a consistent pattern across soil. And also the same pattern observed when we focused on the sole presence of microplastic, where we see again transient and microplastic type specific effects, particularly related with presence of PBAT on the denitrifiers abundance. And these effects also were mostly present in French and Dutch soil in this case, with Greek soil remaining largely unaffected. To wrapping up now, our main results. First, nitrification activity showed strong soil-dependent interaction effects with the clear signals observed in the Greek soils, as you could already realize. It was very obvious. Second, and this goes again mostly for the Greek soil, microplastics alone stimulating nitrification-related endpoints indicated that maybe they could modulate microbial functioning. Third, again, mostly goes to the Greek soils. When co-occurring with albendazole, the stimulatory effect was reversed, leading to functional and abundance suppression. Fourth, and this is a little bit more general, the magnitude of responses on ammonia oxidizers were kind of group specific and time dependent, while the nitrifying bacteria on the other hand remained stable and overall less sensitive than ammonia oxidizers. So taken together, these findings highlight the complexity of nitrogen cycling responses under combined stress. So as an overall conclusion, then our findings suggest that microplastic could act as ecological modifiers rather than inert particles in soils. They can influence both fate of co-occurring stressors and how microbial communities respond to them. And we think that this complexity of interaction cannot be captured by just single stressor assessments. Moreover, the soil-dependent response we observed indicate that ecosystem sensitivity is soil context specific, potentially linked to factors like physicochemical characteristics of soils or soils' baseline microbial diversity, maybe. And at this point, I would like also to mention that the findings we present you today, and of course more of them, are included in two recent MINAGRIS-related publications we made, published in the Journal of Agricultural and Food Chemistry and FEMS Microbiology Ecology, respectively, where you can explore our study in more detail if, of course, you are interested. But our findings, as Eleni also mentioned, were produced under controlled experimental microcosm conditions, which could raise an important question regarding their ecological relevance in larger scales. Besides, agricultural soils are considered dynamic and heterogeneous systems. And that's why within MINAGRIS, we adopt a multi-scale approach, monitoring similar microbial indication also in pot and field experiments to assess if these patterns can persist or change under realistic conditions. And right now we are also integrating the data obtained from these experiments in order, by combining both laboratory findings with field scale observation to aim to evaluate ecological relevance at higher levels of complexity. So, and I think somewhere here I'm going to finish pretty much. As a final take-home message, our results show that emerging presence of microplastics in agricultural soil can lead to multi-stressor interaction with other chemical stressors like pesticides or pharmaceuticals, which could potentially reshape key soil function like nitrification, with potential implication for agriculture and for soil health. Moreover, the strongest interaction effect were observed in a Greek soil, a soil from Mediterranean basin region, which already considered particularly exposed to climate-related pressures. And while our study did not directly access climate stressor interactions, these findings, we think, that highlight the importance of understanding these multi-stressor dynamics, especially in regions facing already increasing environmental challenges. And finally, we believe that studies like ours highlight the need to integrate this chemical stressor with microplastic co-occurrence scenarios into soil health assessment and environmental risk frameworks, which will be essential for maintaining resilient and sustainable agricultural system and practices. So, at this point, I would like to thank MINAGRIS dissemination team for organizing this webinar and giving us the opportunity to present your findings, and also Euroquality for the technical support of the event. And of course, thank you all of you for your attention. And that's all, folks.
Brilliant. Thank you so much, Stathis and Eleni. That was a really interesting talk. And it just underpins the importance of not looking at stressors in isolation, because in reality, most fields are contaminated with lots of different things, from pesticides to fertilizers to other chemicals. So really interesting that we've been able to look at the interactions of microplastics with a couple of those. So please do post any questions in the chat. We are ready for a question and answer session. So Sashki has asked, how was the microplastic dose decided?
The two doses, the low one, which is the 0.1%.
0.01.
Okay. Represents, let's say, based on the literature and also within MINAGRIS project, independent studies, and also from other, let's say, consortium like Papillon. Let's say a convenient, realistic dose level which can be found in agricultural soils. Although these numbers are dynamics, and we are getting new evidences that reshaping this pattern constantly. The higher dose representing a projection of the microplastic dose in soils into the next years, next decade. I don't remember in detail. So it somehow represents a high risk scenario. Pretty much. There are a lot of questions. I hope we could answer to everything.
Yeah,
I'm
no, that's
hearing-
perfect. Thank you. Yeah, that was great. Thank you, Stathis. So yes, it sounds like we use doses that are, comparable against other research projects as well. So Papillon was our sister project, ended last year. And it sounds like their low dose was the same as ours for, which is obviously helpful for things like comparability. So Pierre has a question. Pierre says, could you please describe more in detail what you mean by a co-occurrence scenario? And have you already drafted an example?
Okay, so we decided to go for albendazole and pyraclostrobin because albendazole we have seen from previous studies. It was from a PhD of Stathis that they can occur in agricultural soils through manure. And for pyraclostrobin, it's a really widely used fungicide, so we decided to go for both of them. And also for albendazole, from previous studies, we have seen that it has side effects on soil microbial communities. Yeah, so this is why we decided to go for those two organic compounds.
However, sorry, I think this question goes in how we integrate this concept of concurrent scenarios in policy making and framework, if I get. I mean, Eleni described why we selected these specific pollutants to coexist with microplastic. What we mean is that we should include in the frameworks, let's say, already existing for pollutants like pesticides and antibiotics or veterinary drugs, the parameter of microplastic presence and microplastic levels in these soils, exactly because there are these complex interactive effects. I mean, I don't know if this cover the question, but at least
Yeah,
that's what I had in mind.
That's great, and thanks, Eleni, for explaining why we chose pyraclostrobin and albendazole as our kind of additional pollutants. We might touch on that a bit more shortly, but are the microplastics that you applied, were they applied as pellets? And if so, what size range was used in the experiments?
Yeah.
So were they pellets when you applied?
Powder-like, basically.
It was powder. It was
Powder.
in the size of less than 250 micrometers.
Yeah, perfect. And there's another clarifying question around why pyraclostrobin, which is a fungicide, and why didn't we look at herbicides that are usually applied more directly to the soil?
Yeah, Yes, we based in previous studies, of course, on the interaction of pyraclostrobin, but also we wanted to choose two compounds that they have different hydrophobicity. So pyraclostrobin is more lipophilic than albendazole. That's why we chose that.
Okay, that's excellent. And just to say, I believe there is more information about this on the MINAGRIS website, because quite a few of our experiments looked at these particular co-occurring pollutants. So yeah, there's further information on our website, I believe, and do touch base if there are any further questions around that. Marco has asked, were the Dutch and French soils dried for shipping before the experiment? So were they dried before they were shipped over to Thessaly?
No,
And also, sorry, I want to add that also it's based on the use. I mean, we had the previous question. I mean, we had to choose specific pesticide for the pyraclostrobin. We had also some work in our lab and some, let's say, experience on methods and ways to measure it. And it's also considered a very widely used one, as it is also albendazole in case of anthelminthics, and also known for effects on soil microorganisms. So we wanted, let's say, some compounds that we know that maybe they can have a kind of effect, and they could interact through hydrophobicity, their hydrophobicity with microplastics. So for the soil, I think they send fresh, not dried, not that dried. If you're asking this question.
Excellent. Okay, that's perfect. Thank you. And Miriam has asked, do you have any experience or an estimate as to whether microbes return to their original rates after complete biodegradation of those microplastics that are biodegradable? So I think we're talking more here about biodegradable plastics, and whether once they're fully degraded, does that help microbes to return back to normal? Do we know that yet? I don't think we do.
yes, exactly.
Yeah, I think that's an ongoing research area, right?
We haven't focused on that. Basically, it needs more time to completely biodegrade. I guess 90 days it was not enough for that.
Yeah. And I mean, the challenge in the field is that with a lot of even biodegradable plastics, they would be applied again and again. So there's probably no opportunity to see the plastics completely disappear. So but ongoing research.
Sorry to interrupt again. But again, if I understood the kind of question is if the reason why we are observed some recovery or some could be related to the by degradation, right? So it was something like that. Yeah, we could not say for sure. I mean, we did not monitor, let's say, soil microorganisms that could biodegrade microplastics. I mean, this is there is a part of MINAGRIS initiative that focused on that, but it was out of scope in our research. But I also agree with Eleni that okay, PBAT and start-based plastic we use, which are considered biodegradable compared to LDPE, but they are still considered plastics. I mean, for 30 and 90 days, I don't have a specific, let's say, evidence for that, but I don't think it's generally enough time in order to actually see difference in degradation. But they have differences on their structure, which could translate it in differences on the surfaces that maybe can be used by microorganisms. And I don't know if there is a question like that, but it's a good assist on that. Maybe one of the reasons that we are observing, for example, that was very clear, a different effect, let's say, or possible effect on microplastic between ammonia oxidizing bacteria, let's say, and ammonia oxidizing archaea, could be that between these two groups there will be differences on how easy or difficult they could colonize the surface of these microorganisms, since these groups are known of forming biofilms. And actually, we are having a follow-up experiment where we will use mesoplastic fragments, so we could extract from their plastisphere. So we could we want to try to actually observe if there is enhanced abundance of these microorganisms to the plastic between the soil. Just an extra. It was not an answer on the question, but I don't know. Maybe someone could have this.
Yeah, no, really interesting, Stathis. Thank you. That's great. So Saskia has said it's suggested by other studies that smaller particle size leads to more severe effects. Do you have any views on that? Saskia, it would be useful if you shared those studies, if you have them.
I mean, we cannot say for sure that these effects are we observed are directly linked. I mean, direct or indirect. I mean, one explanation, for example, of the interactive effects of microplastic and alpendazole to microorganisms could be related with what I said before, that, let's say, plastic, it's a very nice house for microorganisms to live on it, but when it's absorbed or interact with toxic compounds, it's like a house on fire. So it has an opposite effect. So, or we know that microplastics, they are, let's say, affecting soil and soil properties just because they are there. I mean, by their actual physical properties and their presence on the soil. But I think that there are some studies and some evidence about direct effect of plastics on microorganisms, but I think that has to do mostly with nanoplastics, which they had even smaller size, which actually could help them enter into the cell of microorganism and actually and directly poses effects. But I don't know if this is the case with the microplastics. I think the size is big enough in order to, let's say, directly enter into the microbial cell to induce direct effects. But I cannot. I'm not very sure for that, telling the truth. I don't know if I covered the question.
Yeah,
We covered, but
yeah, no, I think that was great, Stathis. Thank you. And another question from Miriam. So she says, based on your expertise in microbes, would the community be able to recover if no more biodegradable plastic was applied anymore? I mean, one thing I'd say immediately to that is in farming, you know, there is some kind of, I don't know, I think it's very difficult to move away completely from using plastic or biodegradable plastic because of a trade-off between pesticides and plastic. And so, you know, for example, a lot of organic farmers, they're not using synthetic pesticides, but then they have to rely on a certain amount of plastic. So I think it's always a trade-offs between, yeah, you know, removing plastic completely and using it where it's thoughtful and powerful, while avoiding contamination. But Stathis, what are your thoughts on this in terms of microbial recovery?
okay, I think that I think that mostly goes, between ammonium-oxidizing bacteria and archaea. And okay, I'm not an expert on ammonium-oxidizers. I work with them, but not an exact expert. But from what I know and from I think I can recall, in generally there is reported that ammonia-oxidizing archaea actually are more resilient to changes, and in many cases we observed in under the pressure of external disturbances patterns similar on what we saw, that uh, an early effect, but a recovery, while ammonia oxidizing bacteria are more susceptible and more let's say difficult to go through these disturbances. And also this difficulty, let's say it's bigger because of the competition between these two groups for the same niche. So if one group is finding difficult to adapt and the other one start adapting, it actually takes resources and the ecological space from the other one. So yeah, I will go for ammonia oxidizing archaea as the most easy one to recover than bacteria. Yeah.
Yeah, excellent. Okay, that's fabulous. So we'll do one more question, because I'm sure it's been quite a grilling, lots of questions today, which is fantastic. Thank you, everybody. But one more question from Jane. So Jane says, to what extent had the soils been pre-exposed to the chemical pollutants prior to the experiment? So, for example, were they taken from arable soils, which may have already had some exposure to them? And if so, could the microbial populations in those soil samples carry different genes responsible for the chemical degradation? And could that explain some of the differences?
Okay, so the idea of selecting those soils was to have no prior exposure either to microplastics or any of the pollutants that we were about to use. So we just did the application to the soils that were no exposure on any contaminants.
And as an addition, yeah, so it is what Eleni said. We wanted soils considered pristine regarding microplastics and pollutant. We don't have, although, okay, in part of the study, we also focused on having some measurements regarding microbial diversity on the soil, but not targeting very specifically, let's say, having some metagenomic analysis on degraders. But, and we did not actually, we have a possible explanation that we referring to that also in our paper, why maybe the Greek soil, I don't know if this kind of could answer to this question, was a little bit more more let's say, sorry, I forgot the word, more sensitive to multi-stressor. We observed that here we can see a Shannon index alpha diversity, which shows the basic microbial richness of the study soils regarding total fungi, bacteria, and the protist population. And we see with the orange yellow that the Greek soil, especially regarding bacteria and fungi, was significantly less diverse than the other two soils. And in literature, there is like molecular microbial ecology, there is some kind of trend between higher soil diversity and let's say higher resistance to external disturbances and possibility of let's say buffer these effects. So we cannot say with sure that this is due to that. It's like an extra
Explanation.
not explanation, an extra clue that maybe showing some trends like that. I mean, to be sure, we should having focused a little bit more on that to give an absolute explanation connecting soil biodiversity with the effects we observe. But this is also shows the complex nature of these interactions, which are not decided by one factor, but we have to gather a lot of different elements in order to make an outcome of these effects. Sorry.
Brilliant. Thank you so much. And yeah, it's fascinating the differences in microbial richness between the different soils. So I think we'll close off there. So just to make you all aware again, we do have our final conference happening on the 16th of September in Brussels or online. Please do read more about it and sign up via the link that Deborah has shared. You can also read more about the research presented today in a further link that Deborah has sent. We have recorded this webinar, so it will be going online shortly via our YouTube and LinkedIn channels. A further webinar will be taking place. We are hoping to run a series of 6, so please do stay tuned. To find out about events soonest, to be the first to find out, it's always good to sign up to our newsletter. So that's available via our website. You should be able to see a sign up form. So please do stay in touch. Do drop us an email if you have any further questions. And a big thank you to Stathis and Lagos and our dissemination team today. So big thank you to Deborah and Julienne, who've been working in the background on making this happen. Excellent. Thank you so much.
One last thing before we close, because I don't know if there were also some other questions and comments we did not manage to, did not have the time to deal with that. Here there are also our personal emails, so for more questions or comments or ideas also, we are very happy to contact with you. So if anyone wants to hear it, we can, there are our emails for content. Thank you again also for your attention.
Thank you very much.
Brilliant. And Stathis, please do note the message from Anna saying she'd like to discuss potential collaboration. Her email address has been posted in the chat, so please do reach out to Anna. That'd be brilliant. Okay, all right. Thank you, everybody. We'll end there. Thanks. Bye-bye. Bye.
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 Bye.